White blood cells, as we know, have various types, but the manner by which they destroy foreign invaders differs. Welcome back to Joanne Academy. In this video, we'll be discussing the various mechanisms of action of various white blood cells. Tighten your seatbelt because it's going to be a sweet ride. The neutrophil plays an important role in the defense mechanism of the body. Working with the monocytes, the neutrophil provides the first line of defense against invading microorganisms. The neutrophils are the free cells in the body, and it is the most free white blood cell that wanders in every part of the body. Let's briefly look at the substance found in the neutrophils and the various functions that they perform. The granules of the neutrophils contain enzymes like the protease, the elastases, the metalloproteinase, and the myeloperoxidase. It is the responsibility of this enzyme to destroy microorganisms, and they also contain antibody-like substances called the defensins. The defensins are antimicrobial peptides which are active against bacteria and fungi. For the membrane of the neutrophils, it contains an enzyme called NADPH oxidase. This NADPH oxidase is activated by toxic metabolite that is released from infected tissues. The activated NADPH oxidase is responsible for bactericidal action of the neutrophil. All this substance that is present in the granules of the neutrophils and as well as the cell membrane of the neutrophils make it powerful and effective killer machine. The neutrophil also play an important role in blood clotting by secreting platelet activating factor, which is a cytokine. So this cytokine accelerates the aggregation of platelets during injury in the blood vessel, resulting in prevention of excess loss of blood. Let's briefly look at the mechanism of action of the neutrophils. Neutrophils are released in large number from the blood at the time of infection by the microorganism. At the same time, new neutrophils are being produced by the progenitor cells. All the neutrophils move by dipedasis and are attracted towards the site of infection by means of chemotaxis. Chemotaxis of the neutrophil are made possible due to chemical substance called chemoattractants that is produced by the infected area, and these chemoattractants are able to attract neutrophils to the infected area. After the neutrophil must have been attracted by the chemoattractant which is produced in the infected area, the neutrophil then surrounds the infected area. Not only that the neutrophil will surround the infected area, the neutrophil will also get attached to the infected area. It is the responsibility of the chemoattractants to be able to make the neutrophil more sticky and firmly adhere to the infected area. One neutrophil can hold about 15 to 20 microorganisms at the same time. It then engulfs the bacteria and then destroys them by means of phagocytosis. Another mechanism of action of the neutrophil is known as the respiratory burst. Respiratory burst is a rapid increase in oxygen consumption during the process of phagocytosis by the neutrophils and other phagocytic cells. So the NADPH oxidase is responsible for this respiratory burst. During respiratory burst, the free radical, which is oxygen ion, is formed. Two oxygen ion combine with two hydrogen ion to form hydrogen peroxide. Both oxygen ion and hydrogen peroxide are the oxidants having potent for bactericidal action of the neutrophil. There is a whitish yellow fluid formed in the infected tissue by the dead white blood cell, bacteria or foreign bodies, and cellular debris. This whitish yellow fluid is known as pus. The dead white blood cells are called the pus cells. During the war between the white blood cell and bacteria, many white blood cells are killed by the toxins released by the bacteria. The dead white blood cells are collected in the center of the infected area. The dead cells, together with plasma leaked from the blood vessel, liquefied tissue cells and red blood cells escape from the damaged blood vessels, constitute what is known as the pus. The eosinophils plays an important role in defending the body against parasites. During parasitic infections, there is production of large number of eosinophils which move towards the tissue affected by parasites. In allergic conditions like asthma, the eosinophil count also increases. The eosinophil is generally responsible for the detoxification and disintegration and as well as removal of foreign proteins. Let's briefly look at the mechanism of action of eosinophil. Eosinophils are not completely motile and phagocytic like the neutrophils. Some parasites may be larger than the white blood cells, so eosinophils attack them through cytotoxic substance that is present in their granules. When these cytotoxic substances are being released over the invading parasites from the granules of the eosinophils, This substance becomes very deadly to the invading parasites and then destroy the parasites. Some of the deadly substances that are in the granules of the eosinophils and are released as the body is being exposed to the parasites include the eosinophil peroxidase, the major basic protein, the eosinophil catenoic protein, the eosinophil-derived neurotoxin, and the cytokines. The eosinophil peroxidase are the enzymes that are capable of destroying worms, bacteria, and tumor cells. The major basic protein is very active against helminths, which is worms. It destroys the parasite by causing distension and then the detachment of the skin covering the organisms. The eosinophil catenoic protein is the major destroyer of the helminths, and it is about 10 times more toxic than the major basic protein. It destroys the parasite by means of complete disintegration, and it is also called the neurotoxin. As for the eosinophil-derived neurotoxin, it destroys the nerve fibers, particularly the myelinated nerve fibers. The cytokines such as the interleukin-4 and interleukin-5 accelerates the inflammatory responses and the killing of the invading organisms. Let's wrap it up by quickly looking at the basophils, the mechanism of action of the basophils, and in our next video we'll look at the monocytes and the lymphocytes. The basophils play an important role in the healing processes. So the number of basophils increases during healing processes. Basophils also play an important role in allergy. The reason why basophils play an important role in allergy is because of the presence of receptor of IgE basophil membrane. Now let's talk about the mechanism of action of the basophils. The function of the basophils are executed by the release of substances from their granules, which include the heparin, the histamine, the protease and meloperoxidase, and cytokine. The heparin is an essential anticoagulant that prevents the intravascular blood clotting. The histamine, which is a slow-reacting substance of anaphylaxis, produces the acute hypersensitivity reaction by causing vascular and tissue responses. The serotonin and bradykinin are also inclusive. The protease and the meloperoxidase are enzymes which exaggerate the inflammatory responses. The cytokines such as the interleukin-4 accelerate inflammatory responses and kill the invading organisms. There are some substances that resemble the basophil, and these substances are known as the mast cells. They are large tissue cells that look exactly like the basophil. The mast cells are found along with the blood vessels and are prominently seen in areas such as the skin, the mucosa of the lungs, and the digestive tract. These cells usually do not enter the bloodstream. So let's briefly look at where these mast cells originate from. The mast cells are developed in the bone marrow, but their precursor cells are different. After differentiation, the immature mast cells now enter into the tissues. Maturation of the mast cell takes place only after entering inside the tissue. Now let's look at the functions of the mast cells. The mast cells play an important role in producing the hypersensitivity reactions like allergy and anaphylaxis. When activated, the mast cells immediately release various chemical mediators from their granules into the interstitium. There are two substances that are secreted by the mast cells. The first is the preformed mediators, and the second is the newly generated mediators. The newly generated mediators are substances that are absent in the mast cells during resting conditions and are produced only during activation. These substances are the arachidonic acid derivatives such as the leucotrienes, the leucotriencyls, and the prostaglandins, and as well as the cytokines. The preformed mediators are substances that are already formed and are stored in the secretory granules of the mast cells. This substance includes the histamine, the heparin, the serotonin, the hydrolytic enzymes, the proteoglycans, and the chondroitin sulfate. So guys, I hope this video will be helpful to you in understanding the mechanism of action of the granulocytes, which is a type of white blood cell. So in our next video, we'll be talking about the mechanism of action of the monocytes and the lymphocytes, which are the agranulocytes. Thank you so much guys for watching this video. Please support me by subscribing to my YouTube channel. Share this video with your loved ones, with your friends, so that the information will be able to get to various people. Until then, remain blessed. God bless you. God got you. Peace.